So, hello, welcome to seminar today. Today, the presenter is Michael Miller. He grew up in the Twin Cities and at the University of Minnesota for his undergraduate study. And he is now pursuing a master's degree in Dr. Laura Shannon's lab. And he's using Genomic to assist potato breeders. And fun non-fact about him is that he loves dog skiing in the winter. And thank you. So now, Michael, present. Thank you once again.
Okay, thank you for the introduction, and thank you everyone for attending my seminar. So we'll start with a bit of background on potatoes. So potatoes are farmed on over 90 million hectares worldwide. They're the third largest food crop by consumption and the largest non-crop, and are valued as a nutrient and calorie dense in much of the world. This importance, there's a lot of difficulties in plant potato breeding that has meant that we haven't seen the same progress that we've seen in other important crops like maize. It's an autotetraploid, highly heterozygous plant. So to break down what exactly that means, we'll start with the tetraploid part. Diploid is going to have 2 sets of chromosomes, while tetraploid has 4. And then the auto part of autotetraploid is as opposed to allotetraploid. An example of an allotetraploid plant would be durum wheat, which despite being tetraploid, it has 2 chromosomes from 2 different species. So those sets of chromosomes will tend to pair together during meiosis and kind of treat it like a kid with double the amount of total chromosomes. And you can kind of think of it that way successfully, but that doesn't really apply to potato, as it has 4 sets of chromosomes all from the same species, which means that they might form pairs of 2 like this, or they might form a group of 3 in, or all 4 of them might pair together during meiosis. So that really complicates the just understanding sex of potato, and then also increases the variance that you'll see in potato. An example of this, you can see to the right here, these potatoes that I was planting this spring. This is all one family of potatoes. This is all full that same parents. You wouldn't know that without me saying that, because it's pretty much the full span potato that you can see in this plate here. So that makes it really hard to target a particular offspring from a cross that you'd be looking for. You almost kind of have to just make a lot of crosses and hope that you get lucky with offspring that just works. You can't really kind of build on existing cultures easily. And beyond that, the focus is on many traits beyond yield. This is to some extent true with every crop, but this is especially true with potato, where there's a lot of quality that you have to fit depending on what the end use of the potato is going to be. And those uses form these markets, which I'll explain of here. So this is the market class. These are potatoes are going to be going kind of directly sold to consumers for using as like a side dish and things like that. These probably have the most variety of any market class. You can see 3 different colors here, but there are still requirements that would apply to any fresh market potato. You're to want smooth, bright skin with a bright color of some kind. And like I mentioned, you're going to want it to be smooth skin. Chips, you're probably less familiar with the potato, because usually you'll see just the end product of the chips. Peas, the skin is a little less important since you're really just seeing the slices, but the flesh color has to be a light color and stay that way through frying. A lot of potatoes, if you were to try to fry them, they're going to get a lot in of being fried. And then also the shape is important. You're going to want either a sphere or kind of like a cube kind of shape. Basically, you want a lot of chips that would be approximately the same size to be able to come from the potato. Is the last market class I'll talk about, and probably the most kind of iconic market class. It's probably what you when you think of a potato. And like the previous two classes, you want a long and narrow potato. This is because it makes nice fries that have this effect of flowing out of the container that they're in. So want actually russeted skin, which the market class is named after, which is kind of textured skin. And so within russets, probably the most famous potato in general is the russet Burbank, which was the releases of in 1902 and is still 40% of the potatoes grown in the US. So that's how little progress we've made, that a variety released in 1902 is still representing 40% of it. And part of the reason for that is you can see this russet Burbank on the left. And then you compare, maybe there are a lot of issues with russet Burbank. It's not just that, we made this perfect improvement that created a lot of inputs to much yield from it. So maybe you find this other potato that just produces tubers at basically no inputs. It sounds great, but then the tubers that it's making look like this. That's not going to replace russet Burbank because that shape is not going to make very good fries. People aren't really going to buy it. And so then farmers aren't going to grow because they aren't going to be able to sell it. So that's some of the challenges that we face in new potato varieties. And so with all these problems, we do potato breeding. So if you know a little bit about what a lot of the work that my lab does and just kind of in general what is talked about in potato recently, you might think, well, they're just going to make a diploid breeding system that's going to be like maize, and how much progress we can see in maize over time. So you can see the same thing in potatoes then, right? Well, maybe, but that's really going to be many years from now. So we're just going to look at how we're going to be able to improve the tetraploid breeding system. We're going to do this through new genomic technologies, which are going to require quantitative phenotyping data. And so on the measuring, it's going to be easier or harder to get quantitative data from it. So for example, plant height of this plant here is pretty easy to get a new value assigned to it. You can see somebody just stick by it and you can get a height measurement. You can get a more precise measurement, but you kind of know right away how you can get a numeric value compared to say how exactly these here might be a little bit harder to put in numeric terms. You can say, they're very red or something, but to say exactly how red or how much more red they are than other potatoes might be harder to say. And usually how we've done this is through visual scale. So they're used for many different types of traits. Disease traits are a common area they're used in. Then also quality traits, post-harvest things like skinning, and then color. And they can definitely be useful. They have been used for many years, but they have a lot of weaknesses also. So I'm going to take this skinning scale here that has 4 points to it. If we were to take this top potato here in one of these categories, I think it's kind of in between 1 and 2. let's say it's a 2, and then let's take this bottom one. It's kind of, to me, in between a 2 and a 3. Let's say that one's a 2 also. Well, now you have these in your data as basically, if you look at them, the amount of skinning that you see on the server is not identical. There is definitely a difference. And you can have someone else come in and say, no, I think this top potato is a 1 and this bottom one is a 3. And you can't really compare your data with theirs because you're using a different scale in your head, sort of. And so this is a problem that we'd like to solve through remote sensing and image technologies. So remote sensing is technically just getting without making contact with the thing you're getting data from. So usually we do this through light. Technically our eye are remote sensing devices, and image analysis is getting actual data from the output of those sensing devices. So there's many types of imaging that would be types of remote sensing. So RGB imaging is just like a camera that would be on your phone, or you take normal pictures with. Hyperspectral is looking at wavelengths outside of the human eye can see, and then thermography would be used for something like respiration in a field. But of these, RGB is definitely what people would be most familiar with and what's easiest to get for. So great if we can just use that for getting scientific data from. And in terms of getting that data, I'm just going to talk a little bit about how we would go about getting actually numeric data from photographs. So in a digital image, you're going to have a 3-dimensional array made up of pixels, and each pixel is going to have values associated with it, which are the red, green, blue values. So I have way zoomed in on a spot at the edge of this potato to get the pixels here. If we were interested in the color of these pixels, we could look directly at color values, either all 3 numbers associated with the pixel, or if we're really just interested in redness, we can just look at that. But then we can get information like the shape of an object or how many picture by using the range in these values from row to row and column to column. So I think best in the green and blue numbers for these 9 pixels here. This bottom row is going to be red. It's going to be at the edge of potato. And the values that you see for that are pretty different than this top row around the background. So you can use that big change to indicate that this is the end of that object and the start of the one or start of another object. And so you can kind of take that and to another level using machine learning, which is there's a lot of not helpful definitions. The most helpful I've found is automated pattern recognition. And so it's useful for picking up on more complex patterns that you can see in images. So for example, this perennial ryegrass leaf here is infected with both stem rust and crown rust. And we're able to have a machine learning algorithm on what those pustules look like, are able to find the stem rust pustules and the crown rust pustules, and then give us say the number of pustules or the amount of the surface of the leaf that is covered with that disease. So that's the type of thing that you can use machine learning for in image analysis. So the traits that we will specifically be looking here for this project are going to be color, shape, skinning, bruise, and russeting. Color and shape, I feel like, are pretty self-explanatory. Skinning, we can see in this potato here, you can see the red skin, and then the yellow flesh is the area that is skinned. This is not ideal because and also can be an entry for these that will lower on that potato will last in storage. And then pressure bruise and russetting, I'm going to get to a little bit more on. Pressure bruise, you can see here to the left, it's these grayish black oval or certain spots that will form on potato like 3 to 6 months of storage. And this happens because potatoes are stored basically in big piles on top of each other. And if you've ever had to carry a big sack or a big crate of potatoes, you know that potatoes are very heavy. And so that weight of the potatoes is constantly pressing down on the potatoes near the bottom. And that's going to cause these bruises, which again, don't look very good and can be an entry point for disease. Growing is a little bit more complicated in terms of if you get it or not. It kind of depends on the market class that you're wanting this potato to go into. So for the russet market class, obviously you want russeting, you want it to look more like this, where there's a very rough texture to the skin. But then for fresh market, you want a smoother, kind of shiny surface to the skin. And you don't tend to want textures in between, but you'll see a lot of that during breeding. So you want to be able to kind of quantify this in a scale. So to summarize what I've gone over so far and kind of give rationale for my project, potato genetics make planting crosses and making incremental improvements very difficult. Tuber quality traits are important to the marketability of a potato variety. Visual scales are the primary means of evaluating these traits, but lack the precision required for modern breeding. And then image analysis tools provide the more precise measurements that we need. So my objectives for the project are to create an R package that can evaluate potatoes in shapes, which is called TUBAR or tuber analysis in R, and compare those to visually traits, to utilize machine learning platforms to classify and measure skin characteristics, including pressure bruise and russeting, and then to create genomic selection models for the U of M program. We'll start with that first objective. So kind of beyond that overall objective, have these specific goals in mind in creating TUBAR. I want it to be objective and consistent so that regardless of the time of day who's taking the data, you'll get the same information, the same data from the same picture. I want it to be quantitative. So instead of getting a redness of like 5, you get something like 26.7, and then be scalable. So instead of having to open each individual image, run image analysis onto the next image, which is really kind of defeats point of doing this on a computer, you can get data for a full folder of images. So the population of potatoes I used for this was pretty much all the potatoes I could find, since all the variety that you can see in potato, and really consisted of several different subsamples from several different environments from the lab that were grown at the Sand Plains Research Farm. So there isn't a strict experiment design in terms of which potatoes use, because it's kind of many different experiments that all have their own experimental. And then for pressure bruise, since that isn't something you can just see on a potato coming out of the ground, those were potatoes that were in a chamber at the USDA Potato Research Worksite in East Grand Forks, Minnesota. And also some samples that we got from commercial processors that would be something like a natural case of pressure. And so for taking these images, we use this light box and DSLR camera setup that Krauman at the University of Wisconsin first came up with. It's a very low cost basic setup that's easy to use. We take pictures of 10 tuber subsamples of each clone or plot that we're interested in. And then we evaluate those same clones for scales so that we have a means of comparison with the tuber traits. And so as an example of an image that we get from up, we have 10 tubers here. They don't have any particular order or anything. The tuber is able to identify them from the ground. And we also have a color card here. This is because even within the light box, you'll have fluctuation that makes it hard to get very accurate color data. And so this is used to correct for different light levels that you accurate color data across levels. And then we also put a tag just so that we know what this sample is. So the first category of traits that 2R is going to be looking at is shape traits. And so this is visual scale off to the left, and then 3 metrics are what we're using in 2-bar. So the first 2-bar is measuring for is box or the length width ratio. And so this is basically just looking at the longest and widest line that you can draw across the tuber. And then compactness, taking the perimeter of the potato, and it's going to be comparing that to a perfect circle to get measurement 0 and 1, how close it is to a circle. And roundness is kind of doing that same thing. It's to a perfect circle, but it's not including these little divots and bumps and things of the tuber. It's just kind of overlaying an oval over it. And the reason we take this measurement is because it's going to be really strongly impacted by small, like the soil environment that the tuber is growing in, and might not be that useful for breeding purposes. And color, I talked a lot about RGB scales, but actually we convert that into the lab color scale, which is based on how the human eye sees light. So it's going to have 3 to it. It's going to have a lightness value, A value, which is the red versus green, and then the blue versus yellow value is B. Uses that B value to try to find a point on the tuber where there's a big change in the surface. So for example, here that would indicate going from this red skin to yellow flesh. And so it's going to find that point where there's a big change in the color. It's going to say that the pixels that are above that amount are skinned surface and the rest not. It's going to give a percent surface that's skinned. And so we use these traits and measurements in our visual measurements to on some data that we from a 2018 experiment to get these broad sense heritabilities. The reason that we're using broad sense heritability is because it's a metric that would be useful to breeders in terms of we want a high heritability as breeders so that there's kind of more genetic room to work with in making genetic improvement, because it means there's more variation that's due to genetics instead of the environment. So we're using the same clones and plots to make the ability measurements. When you see an inheritability for the same type of trait, that should be due to our measurements improving. It's a means of saying how much our measurements, our ability to breed. So we see a small increase in broad sense heritability for skin and for color intensity, which is what we call our visual scale, but for 2-bar, we can actually get into redness and lightness. That's part of the bit of image analysis. Sometimes is to break those traits down into smaller factors that are able to be specific. And for sure, you see a really big increase from the visual scale to roundness and length to width ratio, which are almost double the parity, but see a little bit of a decrease for compactness. And I think this is probably due to what I was saying about that being really strongly influenced by small environmental effects in the soil. And so is actually a released R package. It's able to measure color, shape, and skinning currently. It's available on the Shannon Lab GitHub if you want to read the vignette or look at the package. So now we'll move on to the second objective here, which is to use machine learning platforms to classify and measure pressure bruise and russeting. And so how we're going to do this segmentation, and what this is doing is it's basically trying to break the picture into different categories. In this case, we have 3 categories. We have bruised tissue, healthy tissue, and round. And the way that I do this in a program called ImageJ, which is an image analysis program, and then specifically the WECA image segmentation for ImageJ, which WECA is like a collection of different machining, which kind of allows you to look at different ones and see which ones you like better for your particular application. And then this plugin also gives a really nice interface for doing this image segmentation. And so what I do here is I mark area of the image that belong to a certain category. So I say that this here is bruised tissue, this here is bruised tissue, this here is healthy tissue, this background, and I do that for a couple of spots picture. And then I give that data over to the classifier to try to figure out what patterns are seen in the pixels that I marked as belonging to that category. And it then tries to use that information on the rest of the pixels in the image to classify them. And then once I have a classifier that seems like it works pretty well for that image and maybe a few other images, I take that classifier on a different image to at a larger scale. And so that was pressure bruise. Russet, basically the same way. It's just smooth, russet, and background categories is really the only difference. I'm a little bit further along on the russet function. I may actually get a numeric value from those segmented images by using these probability maps, which are just showing the what part of the image belongs in the russet category, what part of the image belongs smooth tissue category. And I may do, well, I have software count the number of pixels in each of those images and then use that to get a print of the tubers that is russeted. And so I'm still working on these pressure bruise and russeting functions. Pressure bruise function, we need a little bit more definition of exactly what we don't want in pressure bruise, as far as is the worst of one big a bunch of small pressure bruises, things like that. It's not a very well studied disorder in potatoes, so we'd like to have a little information from people in industry about what they're looking for when they would reject a potato having too much pressure bruise. And then we'd also like to use this method for identifying other tuber diseases like scab and silver scurf. And over here, you can see very scabby potatoes that don't look good, and then also can have entry for other diseases. So we have this quantitative trait data now, but what are we going to do with it? So we're combining it with high quality genetic data to get to use in marketed selection and genomic selection, which I'm most focused on genomic selection, but I will start with a little bit of an explanation of marker assisted selection, just because I think it will be a little bit easier to understand genomic selection, an idea of how marker assisted selection works. Marker assisted selection, we're using genetic markers that are strongly associated with phenotypic trait. This is useful for traits that can take a long time to be expressed or expensive to measure. So examples of this would be like an apple, where it can take years to get from apple seed to a tree that has apple fruit on it, but you're really mostly concerned about the fruit. So it'd be great if you could find traits about the fruit that are associated with a genetic marker that you can then use on young seedlings instead of having to wait to actually see the apple. And then for traits that are expensive to physically move, an example of this would be disease resistance. So instead of having to set up a whole disease nursery that you have to plan, speculate, and have people rate and things, it would be great if you can just find a marker that's associated with resistance to that disease and not have to do the process of having disease nurseries every year. And this works, it's like disease resistance pretty well, because that tends to be controlled only by genes, but it doesn't work so well polygenic traits that are controlled by many genes. So on the left here, we have a trait that have few associated with it and has this one marker that's very strongly associated with the trait. This would be a good candidate for marker assisted selection, but then trait on the right would not be a candidate because it has a lot of different markers associated with it that are all not particularly high effect size. And this would be a situation you can use genomic selection. So genomic selection is enabled by modern dense marker arrays. And instead of looking for a specific marker that's associated with your trait, you just look at all of the markers across the genome, and you kind of have each of those has a certain amount that they add or subtract a trait, and you kind of add those together to get a genomic estimated breeding value for that genotype. And this works really well for selecting parents in a cross, being able to separate things like dominance effects from the breeding value, which is what you're able to actually pass on to offspring. And this is especially useful in tetraploid potato because you see even more dominance effects when you have 4 different alleles at the same site. Despite it being so useful, it was only made possible in potato. We only have recently gotten SNP coverage and software tools that are able to create genomic models for autotetraploids. So going into going to be with selection, we're again to be making genomic selection models for the University of Minnesota program and using that as a proof for VAR and some new polyploid genomics tools. And so our population for this was our breeding program material in the second, third material for 2020 and third and fourth field year for 2021, which would be the same material, just advanced 1 year. These are from different parents and market classes and genetic backgrounds. We're considering it on the population. This is a single location 2-year experiment. We genotyped this material with 2,000 SNP, 20 SNP markers. I said you need a dense marker array for genomic selection to really be effective. And then we also perform analysis with 2-bar because you also need good quantitative phenotype data. So the Tetraploid genetics tools that I mentioned a little bit earlier are FitPoly and Size. What each of these do, they're both R packages. FitPoly is actually taking raw genetic data and getting kind of actually fixing the output that you will initially get from it, because this is an issue with auto tetraploids that it's hard to really know what the allele dosage is, which allele dosage in a diploid, there's potential allele dosages, either your homozygous marker, heterozygous, or homozygous for another marker. With tetraploid, there's 5 of those states, and so it's harder to tell those apart. And until recently, you couldn't really get reliable genetic data because of that. But FitPoly will provide that reliable genetic data that we can use in genomic selection, and we combine that with our good phenotype data, which is a package for doing selection, auto tetraploids. It accounts for all that complicated that are introduced by auto tetraploids in making genomic selection models, so that hopefully we can have successful, useful models. Currently, we're still phenotyping our harvest from this. So once that phenotype data is available, we'll perform this selection and get genomic models. We'll continue to genotype our second field directions so that we can hopefully continue to use this selection data in future crosses and selections. And if the models aren't useful, we'll have to collect more phenotypic data and then also look at how we're using these tools or the tools themselves to see how these things can be improved to get good models. So with that, I would like to thank Dr. Darren Higginson at the ARS, Dr. Carrie Schmitz-Carley, who now works at our data, Drs. Laura Shannon, Candace Hirsch, which are my committee members, the Endelman Lab at the University of Wisconsin, Tuberosum Technologies in Saskatchewan, and the lab at the University of Minnesota for helping with this project. And are there any questions? So I see one question in the Q&A from Neil Anderson. Can you define low quality genetic data since you're only using high quality genetic data? So as far as marker density, I can't give you specific markers that are like the cutoff, but I mean, you know, in really old genetic data, when you're first using like marker assisted selection, it would be very limited amounts across the whole genome that you even had. And then as far as the allele dose part, the output you will get from like Illumina sequencing, these SNP arrays will be, I don't know, I can't give you like a percent of how many of those markers will be inaccurate, but it will be less accurate than if you run it through FitPoly to get the data. I got another question, which is, are there any benefits of potatoes being tetraploid? So we think that maybe, so if you look at wild potatoes, it's actually very an exception for wild potatoes to be tetraploid. I think at some point tetraploidy was selected for. And so that was probably because when you look at any kind of vegetative material on a plant, it will tend to get bigger with polyploidy. And so the problem that the potatoes were bigger. And while that is when you try to revert back to diploids because of maybe that, but I think really mostly because of gene depression, you'll get much smaller much smaller potatoes. Basically, we think that the gains that you would be able to get from diploid potatoes would outweigh that you see from the tetraploid potato just being bigger. Also, you can potato and you could double the chromosomes to get a tetraploid potato again if it was better. And I've heard of Weka but never used it. Can you touch on the applicability of the software? Is it possible to export a model that future breeders can use, or does it need to be retrained for russetting on each image? Yeah, so this is something that I've definitely worked on figuring out. So you can export a model as I think what the name of the file name is, but there's a certain file extension associated with Weka models that you can import to an instance that I am planning on distributing certain models with 2-bar. You don't need to retrain it on each image. You can just apply the classifier on a new image. If you have that classifier, is that if you are in like a different breeding program than where that was applied, it might, or just using a different imaging setup would probably be the biggest thing. That model might not be that applicable to your data. So you might have to make for your particular application. From James, this looks like an excellent tool for industry and research. Are there applications for 2-bar that would be beneficial to market scale farmers? So it is a fairly low cost setup for sure for phenotyping compared to a lot of things. So I could see it potentially used smaller scales. You could use it just in, if you wanted to do like an experiment within your own fields, I suppose, about using different types of like fertilizers or something, and wanted to compare how that affects the quality of the tubers. You could use it for that, I suppose, if you were a very a very scientific farmer, I suppose. I think that's the questions.
I was curious how much in these images, how much human left there, where like maybe you have a certain feature that you're looking at versus AI being automatic. You just take an image and you have to do any manual adjustment, I guess is what I'm saying, to the images, like tracing around sections. Does that make sense?
Yeah, so at first I am manually just drawing shapes on image to give just an indicator or notice a pattern in it. The idea would be that once I've given it that initial data, it's able to figure the rest on its own, I guess. There's going to be areas, it's not going to be like 100% classified. We're kind of going for good enough and wrong in a consistent way that won't affect data is kind of what you're going for a lot of these machine learning applications. So you could maybe see, and it might be a little bit small on here. See if you can see near the edges, it tends to think that that's pressure bruise along the edge because there's the shadow from the image. I have tried to make sure to show that the shadowed areas, not particular, I have other models trained that are probably better for this, where I really try to get to the edge of the potato to show that that's healthy tissue all the way to the edge to try to prevent that. But you're probably always going to have a little bit of things like that. But the idea would just be that you get to a point of good enough accuracy, kind of.
Follow-up question to that. So then, when you're doing that, do you ever compare the AI to a manual?
Yeah, so that's something like for publication that I've helped it, where we did specifically that, comparing how the AI measured these traits compared to doing it manually, and that's definitely a good way to compare how well the AI does compared to just manually doing it, which in that case, you can say manual's the gold standard because it would just take a long time, but it's hopefully going to be pretty accurate.
Kind of along the same line. Does like lighting affect your image?